morning, everyone. It's my pleasure to tell you a little bit about Envisia Therapeutics this morning. Envisia started as a spin-out of Liquidia Technologies in 2013 with a $25 million Series A led by Canaan and NEA with Morningside, Pappas, and Wakefield Group joining in. And the idea here was to take this print particle platform and see how we could apply that directly to really new products in ophthalmology. So essentially this platform technology is based in the microelectronics industry so that anything you can do on a silicon wafer down to nanometer resolution like you see here can be applied to make very precise drug delivery systems for the eye. So how do we do that? So first we start with a master template, and the image you see here is of our tiny glaucoma implant ENV515, and you can see this template is continuous throughout this sheet, and it's very precise. We can get down to nanoparticles, microparticles, or several millimeters in terms of feature size. And Liquidia has really industrialized this process. So this has been scaled up to meet consumer products volumes and pharmaceutical industry quality standards. So now imagine this master template and pouring a Teflon-like polymer over the top of this and polymerizing that and peeling it off in a sheet. You get something that looks like this. This is a reverse mold. So now we've got a sheet of this mold where we can fill this with essentially any kind of substance. You can fill it with API, it could be polymer, it could be mixtures of each. And we've been using this technique to develop a portfolio of products. For example, glaucoma product that's in phase 2 I'll tell you about, intravitreal drug delivery systems for small and big molecules, and a post-op inflammation program going into the clinic early next year. So now imagine filling this with your formulation and then pulling it out onto an array. You get something that looks like this. So of course this should look very familiar because it's exactly the same as what I showed you with the master template. This can be done in a production mode, even in early development. In batch mode, we're making a few thousand per batch. And for microparticles and nanoparticles, this can be rolled up on sheets and we can make kilograms in a continuous process. So it's highly industrialized and ready to go. But when we started out in glaucoma, we asked ourselves what would be a compelling target product profile to work on here. And we came up with the full efficacy of a prostaglandin, lowering IOP 24/7 for greater than 6 months from a single administration. We would have less hyperemia than topical drops because we'd place it right next to the target tissue, and of course it'd have to be fully biodegradable. So it took a lot of work to get there, and we ended up with these two different clinical candidates, ENV515-3 and -1, essentially a small and a large. But to get here, we had to do a lot of work. We went through hundreds of different sizes and shapes, polymer blends, loadings, even different APIs to end up with travoprost in this format that we could take into the clinic. And we also didn't want to have to wait for 8, 10, or 12 months for extended toxicology to get going. So we came up with a clever design to go into subjects with ocular hypertension that need to have a cataract taken out so they could be dosed for 4 weeks, and at the time of surgery, the implant would be removed and they would go on with their cataract surgery. We'd also get an aqueous humor sample for PK, and we also would dose the fellow eye with Travitan Z to have an internal control. So pretty clever design here. This design is actually going to be presented by Tom Walters on the podium Sunday afternoon. Top line results here. This is median 8 a.m. IOP change from baseline. Again, it's a small study here. These are the results for the small implant. The larger implants were a little bit long for the best anatomical fit, so I'm showing you just the data from the smaller implants. The Travitan Z is in the dashed line, and you see the high dose and low dose in the blue and yellow lines, and you can see Travitan performed as usual, and these two doses performed very well, comparable to Travitan, in some cases even a little bit better. These patients came in with baseline washout IOPs around 25, so very well-run study, very happy with these results so far. In terms of adverse events, the most common adverse events were essentially due to the dosing procedure, so intracameral injection and betadine in the eye. We did diurnals at baseline and at day 25, so here's what the mean change in diurnals looks like. So essentially the high dose ENV515-3 versus Travitan were numerically equivalent with 28% drop in pressure or a 6.7 millimeter drop in IOP, with a low dose coming in at about 20%. So again, a nice result here. We've got more work to do in terms of optimizing for phase 3 and to get the full duration of action set. And some of this data will be presented by Steve Mansberger on Sunday afternoon in poster format. So let me switch gears a little bit and talk about back of the eye. I think we would all agree that having sustained release therapies for intravitreal use is one of the most difficult technical problems to solve right now. And the great thing about the PRINT process is it allows us to work with very fragile cargo, such as full-length antibodies. This picture you see here, these toroid-like microparticles, are an example of highly concentrated bevacizumab particles we made that are 90% protein. We can then use these proteins to do a number of different, or these particles to do a number of different things, including blending them with sustained release polymers. These can be hydrogels, thermoplastics, whatever. At Envisia, we're agnostic to polymers. We just want to use whatever works for our systems. So you can see that even at the end of the day, it's still a high protein content. It's almost 50% protein. And we've taken these with bevacizumab in vivo to show good performance in a rabbit VEGF challenge model at 2 months and clean safety in a pilot safety study in monkeys. This also works very well for small molecules. So this rod-like structure you see here is actually a 6-month linear releasing, fully biodegradable dexamethasone dosage form that we're going to take into DME in early 2017. But we also have a microparticle format that we call mesoparticles, allows for very high loading, and allows us to work with high payload requirements for drug delivery. So the last program I want to touch on here is in post-op inflammation. Again, another example of where we want to take the power of the therapy and put it in the hands of the doctor and out of the hands of the patient. And in this case, we went with a strong steroid. We went with difluprednate because we wanted to get that full efficacy here, which we see in a rabbit model of cataract, where we do a corneal incision and a weekly paracentesis to keep that inflammation going. You see placebo in blue in terms of inflammation scores. Durezol's in purple. That's 4 times a day, every day, topical steroid. ENV905 in yellow is the same drug, but a single administration, much lower dose. It actually performs better than 4 times a day steroids. So we're excited about that. But the kicker here is that we actually see lower drug exposure to the trabecular meshwork when dosed in this format. So we've got a really good chance to have a good safety profile as well. These microparticles you see on the right is just kind of an example, another reminder of how powerful this PRINT platform is. We can really make anything we want. So it's a matter of what's needed in terms of future products. So let me close by saying, we can kick this up a notch even more. We can make regio-specific functionalized particles. We can make antibody conjugated nanoparticles for cellular delivery. All kinds of things are at our disposal with this platform. And I hope you walk away from this talk seeing how Envisia is taking the power of the PRINT platform to make really unique products in ophthalmology, including a long-lasting travoprost for glaucoma, back-of-the-eye formulations for small and large molecules, and an innovative post-op inflammation program entering the clinic early next year. Thank you very much.